And if you take a look at the cross section of the tubule, okay, this is what you get. Once again, I'm trying to get my pointer, and pardon me for the hold up. All right. So, and some of the things I want you to imagine them like a tube. And I'm saying that the tubes are usually small, and medically or clinically, if you look at some of the medical terminologies, you realize that some of the prefixes and suffixes will always give you an idea about what the whole thing is about. So ULE, which is ULE as a suffix, will give you small something. So if you say tube and you say tubule, then that's going to be a small tube. If you have a duct, which is also a tube, and a smaller duct, then that would be ductile. All right, so anywhere you see tubule, it's just telling you there's a small tube. All right, so seminiferous tubules are just small tiny tubes which are coiled within the lobes or lobules of the testis. All right, so within the seminiferous tubules or a small tube, if you please, can you
this one is nice. So maintain it.
Data, can you put off your audio, please? All right, so if you do a cross-sectional view of the tubule, all right, within the tube, you get a baseline cells. Okay, this is the base. So these cells that form the basement, they are all germ cells. Germ cells are the immature cells that can develop to eventually give you the sperm cells. So these are the germ cells. And you see that when you take this picture, for example, there's another layer. This layer appears to be a little bit different from the basement one. And then you move on, there will be another layer, which is also going to be different from this one. And then eventually, as you move towards the lumen, then eventually they become sperm cells. All right. So these are the germ cells. That is the immature pluripotent cells that can develop to produce the sperm cells. Now, so they are there. And in between, you are going to have this huge, large, tall cell, which we call the Sertoli cells. So if you take this as the seminiferous tubule, you have the germ cells, which form the basement, which are always at the base. All right. So they'll be there. And you are going to get varieties of them. So you have the initial one, which we call the spermatogonia, and then they will develop into another type, another type, another type, another type, and finally get you the sperm cells. Okay, so all the developmental stages, the various ones put together become the spermatogenic cells, spermatogenic, because they are the cells that have the potential to become a sperm cells. All right. So, and in between, you have these huge, large cells, tall cells, which you call a Sertoli cell. So, in the seminiferous tubules, you have two critical cells: one, the germ cells, and two, the Sertoli cells. And we're going to talk a lot about the role of the Sertoli cells. All right. We're going to talk a lot about the Sertoli cells. Then you said that if you take this as a tube, and you take this as another tube, you see that there's going to be a space, and this is the intertubular space, which is also called interstitial or interstitium. Take note, this is a tube, okay, and another tube. In between them, there is a space, and that space is intertubular or interstitium. Within the interstitium, so this is also another, this is one tube, another tube, and in between you have all this. So in between them, you have the interstitial cells or intertubular cells. Intertubular is the same thing as the interstitial. So the cells there are interstitial cells. Okay, these cells are special cells. As we move along, we're going to talk about them. They are called the Leydig cells. Leydig cells. So all these things are within the testis. But you see that the germ cells and then the Sertoli cells are within the tubes. And then the Leydig cells are outside the tube, but in between two tubes. And so they are interstitial cells, which we also call the Leydig cells. And these are the cells that produce testosterone. Okay, so you say that the testes produce the testosterone. But if you go inside, you see that, oh, after all, the testosterone are produced by the Leydig cells, which are outside of the tubule. In between, so within the tubule, you have the germ cells. They are going to be developed to get you the sperm cells. And that's why I've said that in the pubertal stages, where the individual is now beginning to develop the secondary sexual characteristics, you know, testosterone will be produced by the Leydig cells, but a lot of them will go into circulation because they are needed to produce the secondary sexual characteristics, including development or growth of the penis. Of course, including the voice breaking, including expansion of your chest, including hair on your pubic area. All those things are secondary sexual characteristics. But once now they gear into sperm production, you see that the Leydig cells are here. They will produce testosterone, but this within the tubule, these big cells produce what we call ABP, that is androgen binding proteins. And you know that androgens, as far as we are concerned, we're talking about testosterone. Okay, the androgens are testosterone. So if you produce androgen binding proteins and the Leydig cells here are producing the testosterone, where do you think they will go? Straight away, they'll move in here because they have the receptors. This particular cell, which are the Sertoli cells, are producing androgen binding proteins, which are the testosterone binding proteins. They are just close here. So they will attract them and concentrate them. And then now they will be used in the development of the sperm cells. So with time, you see that the amount which will now go into the bloodstream will be very, very small. As we move along, you see that the amount of testosterone produced, about 100, 1 in 100 will be concentrated. You call it sequestration. When you say sequestration or sequestered, it means that they are clamped together or they are pulled together. So they will be pulled into the tubule. So there will be very, very small amount which will now go into the bloodstream to cause other effects of the testosterone. So I'm hoping that you are grasping this picture very well and trying to appreciate what it is. So these are the Sertoli cells. They are big cells within the seminiferous tubules. And the other day I was trying to say mnemonics. Mnemonics, M-N-E-M-O-N-I-C. Mnemonics are anything that you develop to help you to remember something. Okay, and I know that in secondary schools, people try to remember the first 20 elements of the periodic table by singing a certain song. Who can share with us how he or she remembered the first 20 elements on the periodic table? Who can share with us how he or she remembered the first 20 elements on the periodic table?
Sir.
Yes, sir.
Hi, Helena. Hi, Helena. Listen to BBC News on Friday night.
Okay. All right. So the hi, Helena. Are you going to write hi, Helena? You are not going to write it, but you are as you sing that song, it helps you to know the hi, Helena is hydrogen, helium, blah blah. Is that what it? So hi, Helena, you are singing, it becomes a mnemonic. It is a song that you are singing or a rhyme that you are just reciting to help you to remember something. So that's what we call mnemonics. It can be a song. Somebody can form a string of words for him to remember, for him or her to remember a certain action. All right. So I'm saying that Sertoli cells, you remember seminiferous tubules. So for example, if you are confused about Leydig cells and then Sertoli cells, which is which? You remember S, Sertoli cells start with S, or there's an S there. So it will go into the seminiferous tubules. So something like that will help you. All right. So that is what I'm saying, that you are talking about the Sertoli cells, which are within the seminiferous tubules. Okay. Leydig cells are intertubular. Intertubular is also called interstitial. Okay. And therefore, the cells within the interstitial space are called interstitial cells. And they produce the testosterone. As we move along, you're going to see that here, the blood is here within the interstitial space. You have blood capillaries there. All right. Now, as we move along, you see a certain phenomenon that will happen. You call the blood-tacit barrier. Who can share with us another barrier, important barrier within the human system, which I just mentioned, blood-tacit barrier. Who can also share with us another important barrier? Yes.
Grandpa.
Yes.
Blood-brain barrier.
Fantastic. Yes, the blood-brain barrier. That's so once you understand how the blood-brain barrier works, it's the same thing here. This is also blood-tacit barrier. So the blood vessels are within the interstitial space, but they will not be allowed to move in here. Okay, they will not be. And remember that the blood provides the nourishment. So wherever they go, the blood that provides nourishment. If blood is not going to go into the seminiferous tubules, then how do they get nourishment? So that's where this cell becomes very special. They are there to provide nourishment for the developing spermatogenic cells. I hope you are following that. We need to understand some of these principles. So the seminiferous tubules don't have blood supply. And you know that blood supply provides nourishment. So how do the cells here get nourishment? So that's why I'm saying that these special cells, the special cells, are there to provide nourishment. Why are we not allowing blood to get a supply here? As we move along, you know that the body system, the cells have 2N. You know 2N. You know the 2N. Hello? The blood, every system, the cells have 2N because every component you have the 23 chromosomes from each partner. Is that not it? Hello? For each cells, it's made up from a pair of chromosomes, one from your father, one from your mother. Yes, that's what happens. Now, but all of us know that sperm cells have 1N. Do you remember that? Because if they should have 1N so that they go and pair up with the another 1N from the egg of the ovary, so that now they also become a 2N. You understand that principle? So you see that genetically the cells here have genetically they're different from the genetic the cells in the blood system. Is that not it? Hello, are you following that? I want this session to be very interactive.
Yes, please.
So you see that genetically, the cells here, the sperm cells here is N1N genetically, whilst the other cells here are 2N. So if you allow the immune system within the blood to get access to this, what do you think will happen? The immune system will see the sperm cells as what? As foreign, because all cells within the body are 2N, genetically 2N. And if you are bringing in something which is 1N, then the immune system will see it as what? As foreign, and therefore will attack it. It will attack it. And that will be an autoimmune something. So you need these special cells as you move along, forms what you call tight junctions. I'm sure you know types of boundaries. So they form the boundaries that are developed by these big cells, Sertoli cells, provide what we call a tight junctions. And once they are so tight, they prevent entry of anything into the tubule, and thereby creating a different environment here. Okay, creating a different environment altogether here for the survival of the sperm cells. Otherwise, so that's why sometimes if there's a break in the integrity of the boundary, what happens is that there's an access of the immune system with the sperm cells, and then they will develop anti-sperm antibodies. Okay, once they develop anti-sperm antibodies, what happens is that they will now be attacking the sperm that are produced, and then they become inactive, functionally inactive, and then that will be a means or a cause for sterility. So these are some of the kind of things that you should understand as one of the basic information. All right, let's move. So please, if you don't understand anything, draw my attention. All right. So now Sertoli cells, as we saw from the picture, they are elongated, oval, tall, irregular color. Just a description. So that if, let's say, those of you who are in histology, if you
Grandpa.
take, hello, sir.
Yeah, please. I want to ask one question on the diagram.
Please do.
So please, I want some clarification.
Yes, please.
Does diffusion, nutrient diffusion happens in the seminiferous tubulus?
Everything that is required within here are supplied by, you're talking about, you know, there could be some nutrients, and yes, that can diffuse. That can, yes, please. Yes, please.
Okay, thank you.
But what is also important here is that once you see that this is talking about, we will do this one in detail as we move along. You see that the germ cells here are 2N, is that not it?
Yes,
the
please.
germ cells themselves. So once we move into spermatogenesis, that's the development or production of sperms, you see that yes, you have 2N sperms within the what do you call it? Germ cells. The germ cells will be dividing. They'll be dividing, and then eventually there will be one cell. So we talk about the shift from the 2N to the 1N. So there's a mix of 2N and then 1N. These sperms are 1N, and then the basement ones, which is the germ cells, are 2N. What I'm saying is that once we go into the spermatogenesis, we will talk about the shift from 2N to the 1N. So everything that goes on within the cell, within the tubule here, is actually controlled by the Sertoli cells. And so that's why Sertoli cells become very, very important. And then you need to understand and appreciate it. Can we move on, please?
Yes, please. Thank you very much.
Right. So you talk about it, the elongated ovoid, blah, blah, blah. We are just, if you look at the picture, you have a glimpse of it. They talk about it so that those of you in histology, if you do, let's say, what we call testicular biopsy. Okay, you do your staining, and then you see a certain structure, and then so this is this slide. Sometimes for examination purposes, they'll give you a slide and ask you what tissue is this. You look at the structure of the cells, and so this is a typical cytosol cell. Therefore, this tissue or this slide should be coming from a testis or testicular biopsy. So that is it. So this is where you are talking about the irregular columnar cells resting over basement membrane up to lumen of the tubule with irregular indentation, blah, 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 with staining characteristics. So you can appreciate that. Then we said that the cytosol cells actually play significant role within the seminiferous tubules. So they are called substenticular cells. And substenticular cells can be the same thing as cystinocyte. Cystinocyte is a site, this is a cell performing sustaining capabilities. Sustaining, so you want to sustain, something is being sustained. All right, so why are cytosol cells called substenticular cells? Why are they also called supporting cells? Why are they called nest cells? So we try to have an idea about why a particular cell have all these names, and they are due to their various roles. Yeah, so they are called substenticular cells because, and like I said, substenticular cell is the same thing as cystinocyte. Okay, wherever you see a suffix CYT, then that is a cell. Okay, so substenticular cells, because they provide nourishment for the germ cells to develop into functionally mature sperm cells within the tubules. I think that's self-explanatory. We said that the blood supply don't go into the seminiferous tubules. So it is the cytosol cells that provide the kind of nourishment that the germ cells need. All right. Now, they are also called supporting cells because they provide mechanical support for the developing spermatozoa. Okay, so the sperm cells are being supported. If you go to the picture, you see that the sperm cells are being supported and they are carried along because this is the cells that are developing. Before they become this, and like I've said, we are going to talk about all the developmental stages. Now, they are held along by the cytosol cells. So they are protecting them to some extent. Okay, they are protecting them up to a point where, so they'll be attached to the cytosol cell up to a point where now they'll be released into the lumen proper. Okay, so up to a point, they will still be dependent on the cytosol cells. So at first stage, are they released to become independent? We'll talk about all those things. All right. So that's why they are called supporting cells, because they provide mechanical. They also called nest cells. And then we all know what a nest will do. So they aid the maturation process. You know how nests aid and support us to develop. I mean, they develop. I mean, talking about how nests aid in our process of maturation from the baby up to, you see, that is the concept. So because they aid the maturation process of the sperm and can control whether germ cells live or die, because they are supposed to support you. Okay, so if you at a point you have too many of them and now you cannot feed them, then what happens is that they will do that kind of some selection as to which ones are you supporting, which ones are you not supporting. So those ones they will not support, they will automatically die. So they have the capacity to determine the germ cells who should die or who should, and we talk about all those things as we progress to the stage of spermatogenesis. So you appreciate why cytosol cells are so important, and you also appreciate why they have so many names. And we have tried to explain what the role they play to be able to get the particular name that we're talking about. Substenticular cells, supporting cells, nest cells. So they are the same. They can be used interchangeably. But I'm saying that the role they play supports the name that they are given. All right. So now let's do a little about the blood-testis barrier. We said that the cytosol cells form tight junctions and create blood-testis barrier, BTB, which suggests that even arterial blood cannot reach the spermatogenic cells. I think that is self-explanatory, as we've said already. Okay, so because of the tight nature, the arterial blood is supposed to supply blood for nourishment. And you remember that we said that the penis, in fact, we also treat erection properly. But we said, whilst we were talking about the structure of the penis, that there is an arterial blood supply and then the venous drainage. Okay. Now, at a point where the arterial blood supply is increased, and then there's a reduction in the venous drainage, then there's going to be an engorgement. And it's that engorgement that creates erection. Now, once erection is reversed, it doesn't stop the arterial blood supply. The only thing is that the rate of flow of blood will be reduced. Why? Because the blood, the penis is still a living structure which must survive, and its survival depends on nourishment from the blood. So blood will necessarily have to flow through. The only thing is that if it is not erect and it is the flaccid or pendular state, then the rate will be so small. It is just enough to provide nourishment. All right. Okay. So we are saying here that here the seminiferous tubules are not being supplied with blood because they need to protect the spermatogenic cells. Okay, and we've explained why they need to be protected, because if they're not protected, the immune system in the bloodstream will see the spermatogenic cells or the sperm cells as foreign because of their genetic incompatibility. All right. So the blood-testis barrier is so important as it protects the developing immature and mature spermatogenic cells from the immune system and ensures a specialized environment for spermatogenesis. Spermatogenesis is the production of the sperm cells. So, like I said earlier, once they are now protected, they create their own environment, okay, which will promote their development. And if there's any break in the integrity, so if the integrity of the blood-testis barrier gets broken, the immune system will start a response towards the unfamiliar spermatogenic cells and destroy them, sometimes causing sterility. I hope this point is also well explained earlier. All right. So the blood-testis barrier functions in a similar way to the blood-brain barrier by separating the testes from the normal circulatory process of the body and prevent the passage of macromolecules from the interstitium into the tubule. It's the same thing, just to tell you that they are functioning in a similar fashion as the blood-brain barrier. All right, so that certain macromolecules will not get access. So that's how come if you come to microbiology, how come certain organisms are unable to cause meningitis, for example, because they don't have the capacity to cross the blood-brain barrier. However, others are able to do so. All right. So that is the important thing. It isolates the germ cells, which are genetically different from the rest of the body, from the circulatory lymphatic system, preventing is the same thing, just to throw more light on all that we've said already. All right. Again, the barrier prevents blood-borne substances or pathogens or antibodies from gaining access to the spermatogenic cells and prevent the potential for an autoimmune reaction to the haploid. You know, the haploid, that's the one end sperm cells. The blood-testis barrier effectively keeps surface antigens on developing germ cells from escaping into the bloodstream and prompting an autoimmune response. It's the same thing that just works to explain it further and further and further. The important thing is that the germ cells, which will develop eventually to become the sperm cells, need protection, especially from the lymphatic system. It's talking about the immune system. You know that from embryology, and then the immune system, immune development, the body goes through a recognition period where it tries to recognize all the cells within the human body. All right. Now, once that happens, the body will not attack itself because the body immune system recognizes its own cells. Okay, that's how come if you introduce even blood from another person, it's the same human blood, but yet there are certain incompatibilities that will cause the immune system of the recipient to react. All right, so the body has gone through its recognition session prior to, remember also that we said that one of the important things about the reproductive system is that they develop or they become functionally effective after puberty. But you see that the process of recognizing self, the body recognizing itself, takes place right from the fetal development. Okay, so it means that the body has recognized itself already. So if something is now coming in at puberty, during puberty, the body's immune system doesn't know that, and therefore it will try to attack it. All right. So it is important that we understand all these things, and our understanding of this one will feed into our understanding of semen analysis, and therefore sometimes you have to appreciate why we do all these things. All we are needed is the semen analysis, but why all these things? But you see, at this point, you are not just going to do the test, but you need to be able to interpret the report scientifically and to be able to support diagnosis and also be able to help the clinician to also manage the patient. That's why you need this broad understanding to be able to be called a scientist, so to speak. All right.
Hello, sir. Can I ask a question?
Yes, please do.
Okay, sir, please, per the mechanism of the barrier, the BTB, that means when it is compromised and it results in autoimmunity, it can also affect the sperm count. So I want to know
Exactly. In fact, it will cause sterility because the sperms must be functional. And what happens is that as we move along, you're going to get into a situation where we get sperm aggregates. Okay, how do the sperm get aggregated? Okay, how do the sperm get aggregated? It means that there's some immune effect somewhere. And how does that happen? So what it means is that if you get, let's say you do see a lot of clumps, the sperm cells clumping, then there's some aggregation. And you know that aggregation can only happen once there is an antigen antibody. Okay, so it tells you that potentially there is a anti-sperm antibody somewhere. Okay, and an anti-sperm antibody will be developed from the same individual. It is not like somebody's sperm going into another person. No, it is the same person whose body is now reacting to its own sperm cells because probably the blood tested bearer may have been compromised. Okay, so that is the how. And how does it get compromised? A whole lot of things. Sometimes the patient may have some surgery, and within the surgery, probably, especially the urologists, when they are doing some surgical interventions, before you could say Jack, probably they have cut somewhere they shouldn't cut, and they won't tell you. Before you can say Jack, the certain aspects of the testicular fluid may have gone into the bloodstream. Straight away, the body will pick it up and then produce antibodies. Okay, so once the body produces antibodies, it's attacking its own sperm cells. And the only thing that can happen is that the sperm cells will not be free to move. Now they'll be aggregated. Okay, we will talk about all those things as we move along. But I'm saying that once we get to a stage where we are talking about sperm aggregation due to anti-sperm antibodies, you know where it is coming from. Is that all right?
Yes, Grandpa.
Okay, right. So this is the Sertoli. We've talked a lot about the Sertoli cells. Let's now just get a summary of the functions, which we've talked about already, though. So they provide mechanical support and supply nutrition to the developing germ cells. This you know already. They provide suitable environment for fostering differentiating germ cells. Like we know, they make sure that they don't allow anything from the blood to come in. So they create their own environment, which is suitable for the development of the germ cells. All right. So active translocation of interconnected differentiating germ cells projected towards the lumen. Like we said, the germ cells will be developing, and they are developing their nutrition, their support, everything is coming from the support Sertoli cells. So they will be linked up to the Sertoli cells, and then it's like the Sertoli cells will now decide when to release them into the lumen. And once you get to spermatogenesis proper, you see all the stages and how and when the Sertoli cells releases them. So they are translocating. It's like, you know that they have the basement. If you cast your mind back to the picture, you have the basement and then another layer of cell. So from each layer, there's a translocation, and those translocations are actually connected by the Sertoli cells. Okay. Active release of mature spermatozoa into a lumen. Okay. Phagocytic disposal of degenerating germ cells and cytoplasm left over following formation. So what also happens is that during the development, and like I've said, once we are going to also talk about the sperm production proper, you see what happens over there. So along the line, and so because the sperms are too many from the production level, there are too many. So of course, some of them will die. But if they die, they must be assimilated. The Sertoli cells must pick them up. Phagocytic, they have that phagocytic capacity to pick them up. And I'm sure all of us know about phagocytosis. So the cells which have died, if they don't pick them up, remember we said that they are creating an environment, a suitable environment for the development. So if you want to provide a suitable environment, why will we allow dead structures to remain there? They are going to compromise the environment. So all the dead, immature sperm cells that will die will be picked up. Phagocytose. They'll be picked up so that they keep the environment clean and suitable for the developing sperm. I hope that point is also clear. And this is a very important function of the Sertoli cell, so that with the Sertoli cell, the environment is always suitable, or the environment is always suitable and appropriate for the sperm production. All right. We also said that Sertoli cells produce or secrete ABP, androgen binding proteins, and I'm saying that androgens, technically here, we're talking about testosterone. So they are within the lumen. All right. So they produce the androgen binding proteins and they become receptors. I hope that point is clear. For all hormones you have receptors. All right. So if the Sertoli cells are producing receptors by way of the androgen binding proteins, then it means that they are there to capture the testosterone which we produce. And remember, you said testosterone are produced in the testes, all right, but they are produced within the interstitial space. And the interstitial space, the cells there are the interstitial cells. Okay, so it is these interstitial cells which are also called the Leydig cells. And it is these Leydig cells that produce testosterone. And once they are produced, they are outside the seminiferous tubules. Remember, we said that seminiferous tubules, once you have one and another one, the in between is where we have the intertubular or the interstitial. That's where we have the Leydig cells, and that's where testosterone is produced. So now the testosterone will now have to be captured within the seminiferous tubules, and this happen by way of receptors, and it is the Sertoli cells that produce special receptors called the androgen binding proteins, which will now capture them. All right, so it is this capture which will now concentrate the testosterone in the seminiferous tubule for the development of the germ cells, or for development of the sperm cells. It is very, very important to understand these principles. All right. So they also secrete testicular fluid. All right. As we move along, you see that the testis is only producing the sperm cells, but the other fluids that make up semen are produced from other places. Meanwhile, the sperm cells, they are within the lumen. They must move. Remember, we said that within the move, as they move, they will move to the straight seminiferous tubules. Okay, the straight portion. From the straight portion, they anastomose, they converge into the red testis. From the red testis, they also converge further into the efferent tubules and so forth and so on. They at this time, they must get some small fluid within which they can swim. Okay, they are not swimming as the way we want motile sperms to swim. Okay, because at this point they are actually they haven't developed the capacity to even become motile. But there must be a fluid they will use as a medium to carry to be transported. Okay, so it is the function of the Sertoli cells to produce some fluid to become a medium through which the sperm cells will move. I won't say will swim, because at that time they haven't even developed capacity to swim. They'll move through that medium. So it becomes very, very important. There's the testicular fluid. And of course, through the blood-brain barrier, they maintain blood testis permeability. All right. Sertoli cells contain receptors for follicle stimulating hormones, which is necessary to stimulate the maturation of germ cells. This is also very, very, very important. So you see that the Sertoli cells, one, they will produce the androgen binding proteins, which are receptors for the testosterone. Okay. Now, they are also producing receptors for follicle stimulating hormone. Remember, we said that, all right, we haven't done that in detail, but I just mentioned it, that before the body will begin of a man, of course, or a boy, will begin to puberty. It is influenced by hormones. And the first hormone is from the hypothalamus. So we have the HPG. And remember the HPG axis. It is a very critical thing to know, especially when you are doing endocrinology. Now the hypothalamus is in the brain, then for whatever reason, mechanism, it will begin to produce gonadotropin releasing hormones, which will hit the pituitary, anterior pituitary. Then the anterior pituitary will now produce gonadotropins, and the gonadotropins are LH and FSH. The LH and FSH are all moving to the gonads, and the gonads here for the male is the testis. Once they get into the testis, you know that in the testis we have the seminiferous tubules, and then outside we have the interstitial cells or the Leydig cells. The Leydig cells are, as you said, are going to produce the testosterone. So they will need a certain hormone to trigger the production of the testosterone, and that is the LH, luteinizing hormones. So over here to Leydig cells and luteinizing hormones, you can link them as a mnemonic. Remember, we said that if you're talking about seminiferous tubules, you talk about Sertoli cells and then FSH, because they all have some S. You will link them together and know that, oh, FSH will go into the Sertoli cells. Okay. Now, the FSH will go to the Sertoli cells because the Sertoli cells also have receptors for FSH. So within the testes, FSH is going to the seminiferous tubules, and then they are attaching themselves to the Sertoli cells because Sertoli cells have receptors. The same Sertoli cells will also produce the ABH, so ABP, which is the androgen binding proteins, which will also be a receptor for the testosterone. So that what it means is that within the seminiferous tubules, for the development of the sperm cells, you need testosterone and you need FSH for the maturation of the sperm cells. So the FSH will now be received within the seminiferous tubules through receptors on the Sertoli cells. It's a little bit confusing, but just get it. FSH and then LH will all be coming from the pituitary, and they will come to the gonads, which is the testes. Within the testes, where will FSH go and where will LH go? I'm saying that link the LH, you know LH, that's luteinizing hormone. Link the L to the Leydig cells, and link the FSH also to the Sertoli cells, so that you can have, and already you know where the Sertoli cells are, and you also know where the Leydig cells are. So that will help you to know which is which. So Leydig cells will go to, LH will go to Leydig cells, and they will now be stimulated to produce testosterone. Testosterone will move into the seminiferous tubules and then be received by the androgen binding proteins, and which will now concentrate them within the seminiferous tubules. All right. Meanwhile, the developing sperm cells also need FSH. So FSH, which is also coming from the pituitary, will also be received by the Sertoli cells, which are already within the seminiferous tubules, and then now sperm production can take place. All right, it's very, very interesting once you understand the principles. All right. So the sequestration of testosterone in the testes results in testosterone concentration, which can be up to 100 times higher in the testes than in systemic circulation. I think I've mentioned this already, that at the puberty stage, now testosterone will be produced all right, because now the hypothalamus has triggered the production of gonadotropin releasing hormones, which will come to the pituitary. Pituitary will now also produce the gonadotropins, which are the FSH and then the LH. So they'll go into the testicles, and then they'll begin to work. The work will result in the production of testosterone. At this point, the body needs testosterone to develop the secondary sexual characteristics. So all the testosterone will produce, about 99% of them will move into the systemic circulation. Okay, good. Now, at the end of puberty, once the body has now reached maturity, then the concentration will be on production of sperm cells. And at this point, the Sertoli cells have produced a lot of ABP, which is the androgen binding proteins, which are within the systemic seminiferous tubules, like I've mentioned already. So testosterone is just being produced back door, just behind the seminiferous tubules. You have the lady cells which are producing the testosterone. So straight away they move into, as we said already. So you see that now there will be no more. If you look at the those in biochemistry, if you look at the units for measurement of testosterone, you see they are, is it picograms or nanograms? I mean, it's small, very small quantities.
Nanogram per mil.
Very nanograms. You see, and you know, nano is 10 to the power minus 9 or so. Is that not it? So very small amount is in circulation. All of them are 10 times that, 100 times that amount is being concentrated within the testosterone. And the concentration of the testosterone is what we call sequestration. You know, in hematology, you have EDTA. It's also called sequestering. You know that sequestering because its mode of action is to capture calcium. All right. So the capture of the calcium will prevent blood clot from clotting because the clotting cascade should be initiated through calcium. So once the calcium has been sequestered, that is captured, then what do you call it? Blood will not clot. So that's how come EDTA is called sequestering. Sequestering because it's sequestering, it's capturing. So we have a sequestration of testosterone, that the testosterone which has been produced is being captured into the seminiferous tubules. Okay, so that is a key element for you to understand. All right. Now the Sertoli cells also play an endocrinological role. All right. By producing the hormone called inhibin, which has a negative feedback effect on the pituitary FSH secretion. You, like I've said, as you move along, you also have to know that, if you are talking about male fertility, you can't just do semen analysis. You'll be doing a lot of hormonal analysis too, because sperm will have to be produced through action of a lot of hormones. And then if you want to assess whether the patient has a problem with production of the sperm or there's a blockage or something, then you, apart from semen analysis, you need other investigative tools. All right. And so you have to know a lot about some of these things. So Sertoli cells, you know, we said the FSH is going to the Sertoli cells. Remember that FS, because of the S, you link it up with the Sertoli. So FSH is going to the Sertoli cells. Now, in endocrinology, there's always what we call negative feedback. All right. So negative feedback is when a certain hormone is being produced and it has achieved its purpose, there should be another thing which will tell the source of the production that no, stop, because we now have enough. Okay. So, and that which is causing it to stop, it becomes a negative feedback. So you will know that FSH is supporting the production of sperms. Now, at a certain point, the production should be regulated. Otherwise, the whole lumen will even be choked. Remember, we have this is already a tubule, a small tube, and we also have a lot of cells which are multiplying and so forth and so on. If you don't take care, the lumen will be choked. So, and we are saying that the environment should be controlled by Sertoli cells. So the environment should be controlled by Sertoli cells through many ways, including the fact that those who are dying, the Sertoli cells will phagocytose them to clean them. Also, there should be a means to stop, because once FSH is coming in, then the sperm production will continue to go on. So at a point, the Sertoli cells should be able to get a certain signal to the pituitary cells, or the pituitary, anterior pituitary, that stop producing more FSH, because I have enough of the sperm cells. And so it will produce another hormone called inhibin, to inhibit. So inhibin will be produced by Sertoli cells, which will serve as a negative feedback to tell the pituitary to stop producing more FSH. I hope that point is also clear, right? So once you understand all these things, once you get to a lab which is well resourced, you can even assess hormones, including inhibin, including FSH, including LH, and all so forth and so on. And once you are doing all this, you know what you are checking for. You know what you are checking for. All right. So we say that inhibin is secreted by Sertoli cells within the seminiferous tubules, and it is to inhibit the production of follicle-stimulating hormone from the pituitary glands, thereby indirectly regulating spermatogenesis. I hope this point is also clear. All right. If I'm going too fast, please you can let me slow down because you need to understand them. Can we move on?
Yes, please.
I hope my people are not sleeping.
No, we are here.
No, please, we are not sleeping.
All right. Good. So now, enough of the seminiferous tubules. We said a lot about the seminiferous tubules and the cells in there, the germ cells, and then the Sertoli cells, and the functions of the Sertoli cells, the important role of the blood testis barrier, and so forth and so on. I think that is enough, at least for now. Then we said that outside the seminiferous tubules, we have the intertubular, which you also call interstitium. Now, that space there has a certain, some cells, which we said they are called the interstitial cells. Okay, they are called the interstitial cells. Now, these interstitial cells are also called Leydig cells or cells of Leydig. So these are the Leydig cells. I think we've mentioned this already. And we say that in between the tubular cells, in between the seminiferous tubules, we also have blood vessels, we have mast cells, we have macrophages, and all those things. They are all there also to make the testes serve as a real tissue. Okay, all right. So the Leydig cells are located in the connective or the interstitial tissue between the seminiferous tubules. This is well known already. The Leydig cells appear as small groups, large, round, or polyhedral, with eccentric nuclei and light-stage cytoplasm. Leydig cells contain receptors for luteinizing hormones. We said this already. So you will link the LH with the Leydig, L and L, so that you know that, oh, so the LH, which cells provide receptors, then straight away you know that they are Leydig cells. And then it is Leydig cells that also produce the testosterone. All right. So the primary function of the Leydig cells is the production of male sex androgen, which is the testosterone. Leydig cells secrete androgen, testosterone, in response to stimulation of LH from the gonadotrophs of the anterior pituitary. LH is also called interstitial cell stimulating hormone. I think if you read some old books, you see ICSH. That's the interstitial cell stimulating hormone. Let's go back a little bit. We are saying that the interstitial cells is the same thing as the Leydig cells, right? Now, the Leydig cells produce the testosterone, but they do so once they are stimulated by the LH. Okay. Now we are saying that so the LH will stimulate the Leydig cells. Leydig cells are also called the interstitial cells. So therefore, when you talk about interstitial cell stimulating hormone, what are you referring to? Let me go back. The interstitial cells are the same thing as the Leydig cells. The Leydig cells produce testosterone, but they'll do so only when they have been stimulated by LH, which is the luteinizing hormones. Okay. We said that the Leydig cells is the same thing as the interstitial cells. So the cells that stimulate the Leydig cells are also called interstitial cell stimulating hormone. Do you get that point? Now, so what will you now refer to as the interstitial cell stimulating hormone?
LH.
LH. Fantastic. So whatever, especially in old textbook, if you see an old doctor who is requesting for, if you get an old doctor who is requesting for ICS, if you get an old doctor who is requesting for ICSH, then, hey, who is this? Clement. No, Clement. 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 Are you sure you are listening to the discussion here? Hey, please don't waste your time on. This politics. If you want to go into politics, fine, do so. But if you are doing politics at this time of lectures of this nature, I'm sorry, you get me disappointed. All right. I don't want a situation. You see, we have taken my time to explain some of these things. I want us to own the program, to own the course, and also to appreciate it, to understand it, so that even at the end of the whole course, you should be able to know that you have gained some knowledge. Yeah, you should be able, apart from the fact that you'll be doing the SEMA analysis. After SEMA analysis, even 10 minutes, you can know how to do the SEMA analysis. But the important thing is to get a certain level of knowledge or information that will make you feel that you are a scientist. And therefore, you can do any interaction with any doctor at any time about a patient whom you are seeing. So please, let us take the discussions seriously. All right.
Grandpa.
Yes, sir.
Grandpa.
Yes, sir.
So in actual fact, if a doctor should request the ICSH,
yes, sir,
it means that doctor wants the levels of
LH.
How do you call it? The LH.
Yes, that's all. That's all.
Yes, to be frank with you, I have seen this test once by a doctor, and we actually didn't know what it was, so we had to refer.
So if for that matter,
I'm
you are
glad.
learning something.
Yes.
Okay, thank you.
Thank you so much.
Thank you. And like say, so now you understand why it is also called ICSH. You understand why LH is called ICSH.
I see. Yes, please.
Yes, okay. Right.
So Bola Pampana.
Clement, will you please off your mic? If you like, you can even leave the platform, because I don't like what you're doing.
Grandpa, I've been trying to put his mic off. It is not happening. So I ended up.
The thing is that the discussion is discussing some political issues with somebody, which to me is not good.
Grandpa, please, can they take him off?
Yeah, I did that. He's come back and I'm still talking at the background. Clement, if you can hear us, please control your mic.
All right. So, like we know already, testosterone is very, very, very important, and it is for promoting formation of sperm cells, for promoting sperm cells production, and also, as we all know, testosterone is not just for sperm; it's also for development of the male secondary sexual characteristics. And we said that this second level of development dominates during the period of puberty, adolescent stage. After the puberty ends, then the promoting formation of sperm cells also now becomes dominant. I hope that point is clear. That's why in the initial stages, I was just saying that now, after puberty, penile growth is unlikely. I think some people, we are just trying to, but what the penis needs for its growth is testosterone. Testosterone will, the amount of testosterone, about 99.9% of them will not be concentrated in producing sperm cells. So you won't get enough for the development of the penis, unless there's some malam somewhere who has some something else that will help the penis to grow after puberty. All right, good. So now we've done the scrotum, we've done the penis, we've done the scrotum, we've done the testis. So we are moving up. We are moving up. And if you look at the anatomy of the male reproductive system, as we move up after the testis, what is there is the epididymis. All right. I mentioned earlier that epididymis, you may see a spelling that is ending MIS. You may also see a spelling that is ending MES. The MES, like we did for the testis. Testis ending with TIS is singular. Testis ending with TES is plural. So that is what it is. And then epididymis to the one ending with MIS is singular, because you know epididymis is a paired structure. All right. So MES talking about it too. If you look at the picture properly, let me see if I have the picture somewhere here. Yes, if you look at the picture, even this picture doesn't demonstrate the C very well. Let me see. Unfortunately, I don't have a very good picture, but it creates like a C of a sort. All right. Here it is like an it's all, but actually it creates a picture of a C. So it is C-shaped, something like that. It's also paired, as you said, located within the scrotum and attached to the posterolateral aspects of each testis. That is that it runs along the back side of each testis. This is just an English, and you can read and understand. It's made of a long, narrow, tightly coiled, highly convoluted tubule, estimated to be 4 to 6 meters long when stretched out, because it's convoluted, like you saw with the seminiferous tubules. You know, you have a convoluted section, and then you have the straight section. When it's convoluted, that's winding, you can stretch it out. All right. So once it's stretched out, it's 4 to 6 meters, and that's a very long thing. Now, the epididymis has a thick, smooth muscle coat lined with pseudostratified columnar epithelium, which includes principal cells that secrete glycoproteins important for sperm maturation. So over here, what you have to know is that the important function of the epididymis is for sperm maturation. Now, we know sperm its development actually takes place within the convoluted tubules of the seminiferous tubules. Remember, when we're talking about the straight seminiferous tubules, we said that over there, sperm cells are not produced. Okay, so it is only within the convoluted section of the seminiferous tubules where sperm cells are produced or developed. From the straight seminiferous section up to the epididymis, it's just being transported. Now, once they are transported and reach the first section of the epididymis, which we'll talk about as the head, then over there, they will need special proteins, glycoproteins, for them to mature. What is its maturation? The sperm should be able to move. All right. Sperm which cannot move is said to be immature. The sperm has been developed all right. It has its all structures. If it cannot move, it means that it's not performing its function. And therefore, the capacity to move is developed within the epididymis. And therefore, once it has become motile, we now say you have now matured, you have come of age. All right. So within the epididymis, the lining of the epididymis produce special glycoproteins, which will now be used by the sperms for its motility or maturation. So the epididymis is structurally divided into 3 gross anatomic sections: the head, which you call caput; body, which is also called the corpus. Remember, we said that when the penis, you have the root, the shaft is also called the corpus. The corpus here, for as far as the penis is concerned, is made up of the corpus spongiosum and the corpus cavernosum. All right, because of that. But for that doesn't mean that the same corpus cavernosum or spongiosum is within the epididymis. All right, so take note. It is called corpus for some other reason. All right. And then the tail, chorda. All of us know that when we talk about chorda, we're always talking about the tail. So the head, the body, and then the tail. All right. And each segment plays specific functions. It's very, very important to know that. So just as we talked about the root, the shaft or body, and then the glans or the penis, that each section has its role to play. All right. The same way, epididymis has a head, the body, and then the tail. Each section has a specific role to play. All right. The head, which is the uppermost or most proximal and largest part of the epididymis, it lies on top surface of the testis. So immediately after the testis, that section of the epididymis is the head. All right. It is formed by the efferent tubules of the testis. Remember the efferent tubules? We mentioned the efferent means carrying away. Efferent tubule. Efferent is tubule. That's a tubule. And a tubule is a small tube. That's carrying away. And remember the testis, the main duty of the testis is to produce sperms. So if a tubule is carrying away something, what is it carrying away? It's only carrying away sperms. So that's the efferent tubules. All right. That will now carry it. Remember, you have the straight testis, immediately after the convoluted tubules, you have the straight seminiferous tubules, which will now form into the ret. Okay, then you'll move on to the efferent. All right. So the efferent is now the part of the testis, which is carrying the sperms which have been produced by into the head of the epididymis. All right. I'm sure if you take your time to read through, you appreciate what I'm saying. So sperms are being produced by the testis. It's being transported through some other tubules, and then they come to the epididymis. So in between the testis and the epididymis, you have the efferent tubules. The efferent is the carrying away. So anywhere, if you see, let's say, efferent arterioles or efferent arteries or efferent venous, well, when efferent is carrying away, so it means that that is the source of something, and it is carrying the thing from that source to somewhere else. Anyway, somewhere to you also get afferent. That's also afferent too, is also carried away as something else to the main source. Okay, right. So that is just to clarify that aspect. So the sperm begin to gain some motility in this region. Remember, we said that by the time the sperms get to the epididymis, they are still immature. Why are they immature? Immature because they cannot move. And one of the key things we expect a sperm to do is to be able to gain motility. All right. So if you haven't gained motility, it means that you are immature or functionally immature. So That is where it starts, the head of the epididymis. Then the body, formed by the heavily coiled part of the epididymis and is attached to the inner side of the testis and extends the length of the testis. Remember that we're saying that the epididymis is like a sort of a C. So the bigger section of the curve is the body. All right. And remember, it's also convoluted. And remember, we said it is about 4 to 6 meters long. So by the time the sperm move from the head through the body, it is like exercising how to, it's like a baby who is beginning exercising how to walk. So through that means the sperm is learning how to move. Okay, so they are still within the realms of maturation. So sperm continue to mature and gain motility in this segment. All right. The tail, the most distal, when I say the most distal, farthest away, and smallest part of the epididymis, sperm are stored here and gain their final capacity. Okay, I think there's something wrong there. So sperm are stored here and gain their final capacity to fertilize an egg, something like that. I'll do the correction. So here we are saying that by the time the sperm reach the tail of the epididymis, they are fully matured. All right, they can move, they can do anything. They have the capacity to be able to fertilize an egg. All right. At the end of the day, all that we want is to produce functionally mature sperm cells. Okay. So at the time that they reach the tail, they have reached that characteristics. The tail region marks the origin of the vas difference. Remember that after the tail, we are getting to another section, which is the vas difference. We also call it a ductus difference. All right. So at the tail end, this is where sperms are going to be stored until ejaculation is prompted. All right. So the tail region marks the origin of the vas difference and transports mature sperm to the prostatic portion of the urethra for ejaculation. So note, sperm cells mature primarily in the head and body segments and are stored in the tail region of the epididymis. I think this point has been made clear. That by the time the sperms reach the tail end, they are just going to be stored. It means that they have completed the maturation stage. All right. And they'll be stored in there until there's ejaculation. All right. Okay. So this is a structure of the epididymis. But you see that, you see your convoluted seminiferous tubules here. You have your straight seminiferous tubules, which will anastomose to give you the red testis, and then will anastomose further to give you the efferent tubules, and then they will now come to the head of the, here, this section is the head of the epididymis. This is the body, okay? And this side is the tail. So by the time the sperm get to the tail end here, they are fully mature. They are fully mature. And once they are mature, they will be stored here until there's ejaculation, then they will push straight away. Occasionally, certain things will happen, and then even without any sexual stimulation, sperms can move a little bit up here, especially if there's prolonged ejaculation. Somebody have not ejaculated for a long time. The sperms are being produced every day, and so they just get accumulated here, stored here for a long time, and then occasionally you see that some of them will push up, and you see that somebody will just be urinating, and within the urine there are sperm cells. It means that that person has stored the sperm for a long time and is not ejaculating. All right. So this is also another way of seeing the epididymis. Of course, your the great seminiferous tubules here. The seminiferous tubules are here all this while. They will produce the sperms. And remember, we talked about the convoluted section, and here is the straight section, the straight. Here, by the time you get here, the sperms, the production takes place here. Here, there's no production. They're only being transported. All right. And they move into the red testes. They move into, you see here, this is an aberrant ductile. Aberrant. Aberrant means what? You can tell me what is an aberrant. Remember, you talk about efferent ductile. But this is an aberrant ductile. What is it? Aberrant is 40. I mean, a ductile which is incomplete. So it doesn't. So if sperms get here, they will just be locked up here, and they can't do it. I mean, they won't go any further. So this, I mean, when you have things which are too many, they're likely to be some fault. If you have, I don't know how to put it, as anything which is producing too many of it. So when it comes to the sperm cells, sperm production, you see that sperm production, there are too many sperms that have been produced. So therefore, there's a tendency for some sperms to be abnormal. Wherever you have high quantities of things being produced, all right, automatically you are going to get some 40, some percentage which is 40. All right, and that's normal. That's why if you come to semen analysis, you talk about morphology. These days, even 4% of normal sperms is normal, because you're talking about, let's say somebody can have a total sperm count of over 200 million. If 200 million and 4% of 200 million, what is it? You see, so when you are producing a mass, too many things, you are likely to have some percentage of being 40. So you have a lot of ducts which are coming out. So you can have some which will just end abruptly, it doesn't go anywhere. And that is an aberrant ductile. So occasionally, some of these things will happen, and it's still normal, unless there are too many aberrant ductiles. And that can only happen if, let's say, there is a testicular biopsy, and then after histological examination, you saw that there are too many aberrant ductiles. So, oh, this person, it means that anatomically there's a problem with the testicular structure, and then something like that. So this is just, by the way, this is an aberrant ductile, a 40 non-functional ductile. And you know that ductile is also the same as a tubule, a duct, and a smaller duct is ductile. The same thing, tube, a smaller tube is tubule. So you can call it a ductile or a tubule, the same thing. All right. So here we are having the head of the epididymis. This side becomes the body of the epididymis. This side becomes the tail of the epididymis. And from the tail, we are getting to the vas deferens. So forth and so on. So let's look at the maturation, the functions of the epididymis. We mentioned some already. We said that it is the maturation site for sperm. It is not the production site. It is the maturation site. The sperms are produced elsewhere. And by the time they get to the epididymis, they are still not mature. So it is through the through the epididymis where they gain full maturity by way of having the capacity to move. All right. So the epididymis is the primary site where sperm undergo physiological changes to become capable of fertilization. So you see, the other slide had capacity to, or capable of, so capable of fertilizing an egg. That is what I wanted to say there. So here, once the sperms pass through the epididymis, then they can be said to be mature. So if you have a lot of immature sperm cells, of course, you talk about epididymal dysfunction. It means that there's a problem with the epididymis and not from the testes. The testes are produced sperms, and then the epididymis is supposed to make sure that they are mature. They gain maturity. Okay, if they are not, if you are seeing a lot of immature sperm cells, so who do you blame? You don't blame the testicles. You blame the epididymis. So that is how things go. The epididymis generates intraluminal fluids that promotes post-testicular maturation of the sperm. Post-testicular maturation here, we're talking about the fact that after the testicles, what should happen to the sperm is about maturation. And then the epididymis will produce all the necessary fluids and glycoproteins that are required for the sperm to become mature. So this is what is happening. The epididymal epithelium secretes glycoproteins, including free L-carnitine, glycerol phosphocholine, GPC, and neutral alpha glucosidase, which are important for sperm nourishment, bringing testicular spermatozoa that are non-motor and incapable of fertilization to functional maturity. What it means is that the sperm, once they have been produced from the testes and they are stored or they go through maturation in this, these are the things, the glycoproteins that are needed for the sperm to reach maturity. So elsewhere in advanced places, if you have semen and there's a problem with maturation, they will test for, let's say, L-carnitine or neutral alpha glucosidase, something. So any of these parameters can be checked. If they check that and there's a problem with that, then it means that the problem is from the epididymis. So you need to be able to, it's like if you do urine analysis, for example, and you are seeing red cells in the urine, you should be able to have an idea about where the red cells are coming from. Okay, so you don't just say RBCs in urine. Anyone can know that the RBCs in urine, but as it takes a scientist to look at the morphology of the red cells in the urine and determine, because the urine is formed from the glomerulus passing throughout then the bladder through the urethra and coming out. Along the tract, anywhere they can bleed. All right. So, and depending on where the red cells are coming from, it can also affect its morphology. Okay. So, for example, we have what we call dysmorphic RBCs. So if you are seeing dysmorphism, hello, are you there? I'm digressing a little bit.
Yeah.
No,
yes,
sir, we
please.
like it. We are there.
Okay.
We
All
like
right. So if you are seeing dysmorphic, and dysmorphism is like abnormally shaped RBCs. Once you're seeing them in the urine, it tells you there's glomerular injury. You see, because the red cells should not be filtrable. All right, but if there's a glomerular damage, there's a glomerular, there's a glomerular damage, then red cells are being forced to go through the glomerulus. And as they go through the glomerulus, their morphology will be affected. The morphology will be affected to the extent that you see like red cells with a bulb, like pear-shaped red cell, like the poikilocytes of a sort. If you see them straight out of the way, you know that these are red cells coming from the glomerulus. And once they come from the glomerulus, there's glomerular injury. So you don't just say red cells. Yes, you say red cells, but you can say this red cell look dysmorphic, which is suggestive of glomerular damage. So that is that. And in that case, you see huge proteins in there, because once the glomerulus is now affected, protein filtration will be high there. So you get a lot of proteinuria and then dysmorphic RBCs. It's not red cells, that will suggest schistosomes. No, it will not. That will not happen. You understand? So what I'm saying is that coming back, if there is a problem with infertility, okay, male infertility, and look at semen, you can look at certain parameters because you must know if there's a problem, you must know where exactly the problem is likely to be. All right. As you move along, for example, you need to the seminal vesicles. Seminal vesicles produce fructose that serve as the carbohydrate source for the sperms. So anyway, you want to check your fructose level in the semen. So if the fructose level in the semen is reduced, what will you say? It means that there's more function of the seminal vesicles. You don't say that the whole entirety of the reproductive system has a problem. No, it can't be. So that now you can pinpoint where the problem is coming from. So what I'm saying is that if you are seeing a lot of immature sperm cells in this thing, yes, it will suggest to you that there is epidermal malfunction, and then you can to be more specific, you can now do a neutral alpha glucosidase test, which is done in most places. It is done. Once you do it and it is low, then confirm that there's a problem with the epididymis. There's a problem with the epididymis. So such a person, if you have andrologist, a clinical andrologist who is treating this patient, you would not target the epididymis and not the whole reproductive system. That's what we're talking about. Thank you. So the epididymis is a storage site for sperm. The epididymis is considered as a holding tube. Remember, we said that the sperms will be produced from the testes, they will be carried through, then from the head of the epididymis, they begin to go through maturation. Maturation, the head size will be reduced to some extent. If there's an excess cytoplasm, it will be reduced. If there is the tail is not moving, it will try to move and so forth and so on. All those things will happen. And they will happen from the head to the body. By the time it gets to the tail end, they are going to be stored. And once they are going to be stored, it means that they are holding on to the sperm. So in effect, that tube can be described as a holding tube. So sometimes you must understand some of the terminologies that are used in the male reproductive during male reproductive sessions. You see, so a holding tube, as far as the male reproductive system is concerned, is talking about the epididymis, because that is the tube which is designed to function to store the sperms which have been produced. All right. And can take up to 14 days for the sperm to traverse the epididymis. You see, it can take up to 14 days for the sperm to traverse the epididymis. Remember, we said that the epididymis is even the convoluted section. Once you stretch it out, it's about 4 to 6 feet meters. 4 to 6 meters is not a small journey, and yet so it will take them about 14 days. To take about 14 days for the sperm to listen. So what it means is that if you do regular sexual intercourse, what happens is that you don't allow sperm to mature significantly. Okay, because you have a store, right? But if you do regular ejaculation, then your store will be. It's like when you're having a hemolytic crisis. Reticulocyte count is supposed to be a blood reticulocyte is supposed to be up to 2%. Those of you who are hematologists. But if you have hemolytic crisis, then retics will be pushed into the bloodstream, even though they are supposed to have been retained for some time. But there's a crisis. Red cells are being broken down. So retics, which are not fully mature, will still be pushed into the bloodstream to go and support. All right. So that is what we are talking about. If you have regular sex consistently, what happens is that the stored sperms in the tail of the epididymis will have been exhausted. So what it means is that those ones which are in the body, which are supposed to go through full maturation, will now be pushed into the distance. So you get a lot of immature sperms. All right. So you do these things with the understanding that if you have any problem with your semen analysis, you should be able to determine where exactly a problem is likely to be coming from. Okay, that is all that I'm talking about. All right.
Okay.
So,
Grandpa.
hello.
Hello, Grandpa.
Yes, sir.
I wanted to find out the time it takes for the sperm cells to be produced in the, how do we call it?
We'll deal with that when it comes to
okay.
When it comes to we are going to do spermatogenesis, sperm production itself. So this information is just to help you. So that once you get there, some of the things, once you mention them, you can appreciate. Sure. Okay, thank you very much. Yeah.
Okay.
The epididymis functions as a storage reservoir for sperm and protect them from harmful. Yeah, I think we have mentioned that. Epididymis, final lesson is they transport the sperm. The epididymis absorb excess fluid released by the testicles to help move the sperm through the reproductive tract. The epididymis facilitates the transport of spermatozoa along the duct. I think this thing you said already. Then you go to a vas deferens. So gradually you are moving from the testes to the epididymis to a vas deferens. After a vas deferens, I think you end there. Then you are going to do the accessory organs, which is the seminal vesicles, the prostate, and so forth and so on. All right, so let's quickly go through the vas deferens. Now over here too, you can talk about vasa differentia. Remember, we said that a lot of the structures are paired. Some are single. All right, so those that are paired, the spelling can sometimes cause you confusion. And that's why I try to put the plurals also into perspective here so that you don't mess up. So vas deferens is the single structure. Plural is vasa differentia. Okay. And then we said that the vas deferens can also be called ductus deferens. Vas is talking about vessel. It's the same thing. Also as a duct. As we talk about a tube and a tubule or ductus, they are the same thing that can be used interchangeably. All right. So vas deferens or ductus deferens is the same thing. And we talk about those ones if you are talking about one, which is single. Where it is plural, for the vas deferens become vasa differentia. And then for the ductus deferens become ductus deferentis. Okay. Also called the sperm duct. And this one, the vas deferens, is a component of the spermatic cord. Remember, we said that the spermatic cord, the testis develops from the abdomen. When it is coming, it's not just drops just like that. There is a muscle that holds it and pushes it through the inguinal canal. You know the inguinal canal? That is how do you, who can describe inguinal canal for us? You know your I don't know, between your thigh and then your pubic area, that side. Okay. You know why a lot of men have inguinal hernia? Can somebody tell us why men have the proportion of inguinal hernia in men is high? And no, hernia is just a weak section of a system. All right. So, like I said, the testes develop from the abdomen, and once it's some about 2 months up to delivery, they'll push the testes will be pushed with a tuft of muscle through the inguinal hernia and then drop it into the scrotum, which is outside. Once that happens, it's like the inguinal canal area has been compromised. You see, because you have to push through, create a hole there. Once you create a hole there, it will have to be sutured, not sutured as to, I mean, it will have to heal. So once it heals up, with time, it can weaken. All right, a place which has been cut off and then healed, it's still weak. So if the unexpected happens, if let's say you put too much pressure on that area, which is already weak, then it will open up. Once it open up, then intestine. I mean, the intestines is also always weighing on the abdominal walls. So, and that inguinal area had been compromised because that is where originally the testis pushed its way and created a hole. Okay, so that hole which healed up with time, like I've said, can become weak, and then the pressure from the abdomen, from the intestines within the abdomen, will now push it to get more open. So once you get open, the intestines will push in there. Okay, and that is all about hernia. All right, so once this thing is coming, the abdomen is coming, it comes with a muscle. Remember, you talk about the cremator muscles. All right, when you're talking about the scrotum, we talk about cremator muscles, and we also talk about the dartus muscle. The dartus muscles are subcutaneous. When you talk about the skin of the scrotum, immediately after the skin, you have the dartus muscles. The dartus muscles will contract or relax to expand or to reduce the surface area of the scrotum. You remember that in that lecture. So once the dartus
Yes.
muscle contracts, if it contracts, it will reduce the entire surface area of the scrotum. Once you do the entire surface area, what is that doing is that it is avoiding excessive release of heat. Okay, so that heat can now be controlled. Under what condition will that happen? That will happen once the environmental temperature is low. Once the environmental temperature is low, the scrotum will not have to release more the extra heat that it has. So it has to conserve it by reducing its surface area. The same way, at the same time, the cremator muscle, which was the mother muscle, which guided the testes from the abdomen into the scrotum, that muscle is still intact. It is that muscle which is also has a lot of vessels, arteries, and veins, and all that kind, including the vasa deferentia. So the spermatic cord is different from the sperm duct. The spermatic cord is that whole entity, that whole mass of muscle, which is the cremator muscles, plus the vessels, plus everything, including the vas deferens, which we call sperm duct, inside. And we said that during the thermoregulation of the testicular temperature, at the time that the dartus muscle is contracting to reduce the surface area of the scrotum. At the same time, the cremator muscle is also contracting. As it contracts, it is pushing the whole testicles closer to the body, so that it will also become warm. So in a cold environment, you nearly sometimes your whole scrotum and testicles appear to be missing, because it has contracted and pulled it very close to the body. And then that the ductus has also contracted to also squeeze the surface area to its smallest possible structure. All right. So those are the things that you must be guided. And I'm bringing this one because a lot of people confuse sperm duct with spermatic cord. Okay, spermatic cord comprises the entire muscle, plus the blood vessels, arteries, and veins, and then the vas deferens, which we are calling the sperm duct. All right. So you have to get that understanding very well, and that is our concentration for now. The vas deferens. So the vas deferens is a Latin word meaning carrying away. Okay, and it's a vessel carrying away vessel. And the ductus deferens is also a Latin word meaning carrying a duct. So the vas is a vessel, and then the duct is also ductus is also a duct, as I've explained earlier. All right. The vas deferens is it's a long, thick-walled muscular tube that serves as a conduit for sperm travel from the epididymis up to the where mature sperm are stored prior to ejaculation. Right. I think these ones I've explained already, and so the vas deferens begins at the tail of the epididymis. I'm sure this one is sure, and then it will move up to the area of the prostate, and before it gets there, it enlarges. The vas deferens. Take note. At its end, it is taken up, or it is beginning from the end or the tail end of the epididymis. Remember, epididymis, we said that after the testicles, from the efferent ductus, it gets into the head or the beginning of the epididymis, which is the head. It moves into the body of the epididymis and then the tail. And then from the tail, it is now taken off as vas deferens. Okay. Now, so the vas deferens will also move up and then now get to the area of the prostate. Before it gets to the area of the prostate, it will enlarge. No, it says it's a tubule, a small tube. Then it becomes a bigger tube. Over there, that enlarged portion is called ampulla. I hope that point is clear. That enlarged portion of the vas deferens getting to the area of the prostate becomes enlarged. It's called an ampulla. So the ampulla of the vas deferens. You can get ampulla of something else. Ampulla is a tube which is moving, and at a point it becomes enlarged. Okay. The vas deferens is responsible for carrying sperm from its storage place in the tail of the epididymis to the ampulla and to the ejaculatory duct and eventually to the urethra. So as we move along, let me see if you can get back to the, no, I don't have the initial pictures of the male reproductive system. I don't have it on this slide. Otherwise, you see that from the ampulla, you are going to see the vas deferens. Okay, so the vas deferens has now enlarged to become the ampulla. And then you are going to see the seminal vesicle, which is also coming in. The seminal vesicle is also a gland of its own. And every gland will have an outlet, which we call the duct. So the duct from the seminal vesicles will come to join the ampulla of the vas deferens. Are you getting it? The vas deferens has now enlarged to become an ampulla. And then the duct from the seminal vesicles is also coming to the prostate area. So they will now merge. All right. Once they merge, they form the ejaculatory duct. All right. So once we get to a picture, I'll explain that further. But we see the vas deferens. The end of the vas deferens is an ampulla. It's an enlarging area. And then that area will now also merge with the duct, which is coming from the seminal vesicles. And then the two of them having merged become the ejaculatory duct, which will now open into the prostate. All right. So the vas deferens is considered to be an excretory duct of the testis. You know, excretory duct. A duct is just a tube. and it's like if you consider that the seminal vesicle, sorry, the testis has produced the sperms stored in the epididymis, and the vas is now carrying it away and going to throw it, virtually going to throw it outside. Then the vas deferens can be said to be an excretory duct. That's what we're talking about. All right. There are two ductus deferens. Remember, we said that it is either vasa differentia or ductus deferens. Identical in structure and function, which emerge from the two epididymis. Each of the two ductus deferens terminates into the ampulla of the ductus. I think I've explained this, that the end of the vas deferens or duct deferens enlarges to become an ampulla. All right. And then they can all be used as storage chambers for the semen. Remember, we said that at the epididymis, the tail end, we have sperms being stored there. And they'll be stored there until ejaculation. But sometimes when we have a lot being produced every day, and they will pile up. So occasionally they'll be pushed up the vas deferens, and then as they push out the vas deferens, they will end up in the end side of the vas deferens. And remember that end side has also expanded to become the ampulla. Okay, so it means that they can accumulate little more of the sperms that have overflown. Okay, so it means that place can also be a storage area. All right, that can also be a storage area to some extent. So the ampulla acts as a storage chamber for the semen and contribute secretions, blah 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 blah. So that is that. So sometimes you may not have reached orgasm, but you can release as you move along. You have what we call the pre-ejaculate. The pre-ejaculate is that you haven't ejaculated, so to speak. You haven't reached orgasm, but there are some initial lessons that are coming up, some initial fluid that are coming up even before you ejaculate. Those ones naturally they shouldn't contain sperms. But because, like I've said, if you have had somebody who had not ejaculated for a very long time, some of the sperms will have been pushed through up to the ampulla area. And at the ampulla area, they are close to in fact, they are almost within the prostate. And they can find themselves out easily. Okay, and so you can before you can say, Jack, any small amount that you get out can impregnate somebody. You won't say that, oh, but still, you could do in the body, also pregnant. That can happen. So those are small things that you should know. It is true. Sometimes, even though some ladies can be very funny, but this thing too has been proven that you may not have ejaculated, but the pre-ejaculate is enough to continue. All right. So there are other additives such as ergotamine, fructose, nutrient to keep the sperm viable. So wherever you need sperm have to be stored, then there should be a form of nutrition for them. So once you are saying that the ampulla area can store sperms, it means that there should be some nutrients to support their survival. All right. The vas deferens anatomy. This thing you can just read them can be divided into several anatomical portions. These are the epididymal portion, spermatic cord portion, pelvic portion, ampullary portion, ejaculatory duct portion. What it means is that this is a long duct, a long tube, and therefore they can be segmented, just as we had with the epididymis, that you can have the head, the body and then the tail. So the same thing, the vas deferens, which is a long tube, you can have various portions. The epididymal portion is the portion that actually connects with the epididymis. Remember, we said that after the testes, you come to the epididymis. And then the epididymis at the tail end, you get a vas deferens. So that section that joins the epididymis is called the epididymal portion. It is actually a portion of the vas deferens, but it's actually joining the epididymis. Then the spermatic cord, that is the part which is actually part of the spermatic cord. Remember, we said that spermatic cord comprises the cremasal muscle that pulled the testes from the abdomen through the inguinal canal into the scrotum. So that bunch, all right, that bunch of muscles, and then the vessels and everything combined form the spermatic cord. So within the spermatic cord, you also have the vas deferens. All right. So that section of the vas deferens, which is passing through the spermatic cord, is called the spermatic cord portion. And remember that we said that it will now move up to the area of the prostate, and then the prostate lies in the pelvic region. All right. So that's also the pelvic portion. And remember, we said that at the tail end of the same vas deferens, it will enlarge, which we call the ampulla. So that's also the ampullary portion. And remember, we said that once the ampulla is formed, the ampulla now merges with the duct, which is coming from the seminal vesicles, to form the ejaculatory duct. So that also becomes the ejaculatory duct portion. That is all that this slide is talking about. So the same vas deferens, but you can have different portions of it. But the good thing is that the vas deferens transports the stored sperm from the epididymis into the ejaculatory duct and then into the prostate for what do you call it? For exit. All right. So this is just to throw more light on what all that you said. This slide, ampullary portion, ejaculatory portion. Okay. So very good. I think this slide will help me. So we have so here you can see the C nature of the epididymis. Okay, it looks like a C, which is covering the testes, and then you have the vas deferens here. All right. So while the vas deferens comes up here within I know that this whole area is within the pelvic region. Okay, so this is the pelvic portion. Now here you see that the tube has enlarged. So this is the ampulla of the duct. Now the ampulla of the duct, and this is the seminal vesicle. So the seminal vesicle is also a gland. All right, a gland will produce secretions. Secretions must come out through a duct. So this is the duct. So the ampulla and then the duct coming from the seminal vesicle will merge. As they merge, they form the ejaculatory duct. And the ejaculatory duct will now, this is the prostate. This whole thing is the prostate. All right. So the ejaculatory duct will now empty into the prostate. All right. So, and now from here, you are going to get your urethra, which is also carrying everything out. So it is very, very, very interesting. So that is all about the vas deferens. And there's more pictures for you about the same thing that we're talking about. So over here, you have this becomes your spermatic cord. This becomes your spermatic cord. Okay. So you have what do you call it? Yeah. The arteries and veins, and then the sperm duct, which is the vas deferens itself, all combined, everything here. All right. So vas deferens functions, the significance of it is mainly for transportation of sperm cells. That's all. Just to transport sperm cells. And then also mixing with seminal fluid. Remember that, okay, as we move along to get into semen production, you will see that seminal vesicle is producing a lot of fluid. And then the vas deferens is bringing along the sperm cells. So they have at the ampulla, you are going to have a merge with the duct of the seminal vesicles, which is also bringing its fluid, seminal vesicular fluid. So at the ejaculatory portion, you are going to have a mixing up. So the vas deferens is part of the mixing up because it does bring in the sperms. This one is bringing the fluid. So it will mix up so that it becomes homogeneous, something like that. All right. They have control of sperm release. You can read this one, you can understand. Peristaltic contraction, you see, especially during ejaculation. And when you are doing ejaculation, you see that ejaculation comes in fractions. You say it is fractionated ejaculation. So there's peristaltic contraction. Peristaltic contraction is when contracted, release. So it means that in that way, semen does not flow through. It's like when you are ejaculating, it doesn't flow through as if you are urinating. No, it will come in pulsating forms. They're like pip, pip, pip. So the semen will come in that kind of spurts. You call it spurting, S-P-U-R-T. It comes in spurts just because of the peristaltic contractions of the vas deferens. It will contract and release, so that the semen will just be coming in that kind of fractions. All right. So you will end here, and then our next lecture will take us to the accessory structures, which are the seminal vesicles, the ejaculatory duct, the prostate gland, bulbourethral glands. Then from there, then you see that we are gearing up. Thank you very much, ladies and gentlemen, for your time. You'll get a slice tomorrow, and then you read through. I'm encouraging all of you to try and take the lectures seriously, ask questions, and then don't just rely on the slides. I think the discussion should help you more than the slides. All right. You can ask questions. I want you to ask questions so that through the question, a lot of things will come up which may not be captured on the slides. On that note, we bring the first week lectures to a close, and God willing, we start second week on Monday.
Thank you, Grandpa.
You're welcome, sir.
Thank you.
All right. If there are no questions, we call it a day. Bye-bye.